Hi, everybody. My name is Ian McLaughlin, and I have a PhD in neuroscience. And today I wanted to talk about another really impressive innovation in brain-computer interfaces. And so I've talked about brain-computer interfaces several times now, and everything from the predictably famous Neuralink. Here we go. Right, Elon Musk's company, which is, by the way, a bit of a drama-laden story, all the way to a device called BrainGate, which has demonstrated, perhaps the most tangible progress. And I have links to both if you'd like to check them out. Just let me know. Now it looks like there's something going wrong here. Yeah, I wonder why it's failing. Let's try this one more time. Boop, boop. Start failed. Let's see. Maybe that'll work. Anyways, if it doesn't, that's okay. So both of these companies have produced some pretty impressive and fun demonstrations of their technology. Both of these, and they're really worth checking out. But neither of them were focused on what this latest group has demonstrated: helping people who are blind to see again. And so, in short, Neuralink, at least thus far, has demonstrated an incredibly impressive miniaturized device that can be implanted and wirelessly listen and report the activities of neurons in the part of the brain that's associated with how a pig uses their snout to sort of probe their environment, poking and snuffling around their environment, right, while they're exploring. BrainGate, earlier this year, which is a collaboration between scientists and physicians at Brown and Stanford universities, as well as Massachusetts General Hospital, some of the top biomedical institutions in the world. So BrainGate was able to both wirelessly record the activities of neurons, but could also deliver stimulation, and all wirelessly. So it's really impressive stuff. But neither Neuralink nor BrainGate had entered the realm of visual perception, easily one of the most, if not the most, complicated component of our sensorium. It's kind of silly to try and rank how complicated various aspects of our nervous system are, right? But I think it's pretty uncontroversial to suggest that vision is certainly the system that at least dominates our sensorium. We rely far more on vision than smell or taste or touch or even our ability to hear to get a sense of what's happening around us, right? And hey, Daryl, good to see you join in. I imagine this would work for certain types of blindness. Correct. And so we'll get into that. But, of course, that totally makes sense, right? It's the only sensory system, vision is the only sensory system that's updated by changes in our environment that literally happen at the speed of light, right? Our olfactory system operates at the speed of how long it takes molecules to basically spread out through the air around us, which is pretty slow. Our auditory system, our ability to hear, is updated quite a bit more rapidly than that, right, at the speed of sound. The speed at which waves of air molecule progression or compression propagate through the gas around us, right? And then those waves hit our ear balls and then vibrate these tiny little hair cells inside of our heads, faster than the olfactory system for sure, but certainly quite a bit slower than the speed of light, because pretty much everything is slower than the speed of light. And so, obviously our nervous system does not operate at the speed of light, far from it, in fact. And this has been tested and quantified, believe it or not. So to be clear on this, right, while the photons that splash up against the stuff around us, or are sprayed out at us from the various devices and screens that we use, while those photons are traveling at the speed of light, our nervous system operates at the speed of biology, right? Which, to be clear, is quite a bit slower than the speed of light. And so there's a fun example that the neuroscientist David Eagleman uses in his book, Incognito, which is a fun book to read. Definitely recommend it to anyone who's just generally interested in, kind of like light, not too technical or jargony exploration of some neuroscience, right? Anyways, here's a fun example of the relationship between the speed of photons and our nervous system. And so it takes about a half second for our conscious awareness to kind of grok that something has changed in our environment. But we humans are capable of responding to certain changes in our environment more rapidly than this time limit, right, would presumably allow. And so one of the fun examples that Eagleman uses is hitting a baseball. So Eagleman, at least in my humble opinion, is a lovely writer, but I'm not just going to recite his whole example, which is on page 8 and page 9, just in case you're wondering, right in the beginning. But so he highlights that there was a fastball thrown by Nolan Ryan, the pitcher, which was clocked at just under 101 miles per hour. So as a result, the baseball crossed home plate in 0.4 of a second. And so 0.4 of a second is like pretty seriously fast, right? Faster, in fact, than our conscious awareness is actually physically capable of operating. And so, as Eagleman writes, quote, "This gives just enough time for light signals from the baseball to hit the batter's eye, work through the circuitry of the retina, activate successions of cells along the loopy superhighways of our visual system at the back of our head, cross the vast territories to the motor areas, and modify the contraction of muscles swinging the bat." And then Eagleman gets to like the main point of this example. So, quote, "Amazingly, this entire sequence is possible in less than 4/10 of a second. Otherwise, no one would ever hit a fastball. But the surprising part is that conscious awareness takes longer than that, about half a second." So in other words, any batter who's capable of hitting a fastball at these speeds is reacting using components of their nervous system beyond just their visual system, right? If they were only using their visual system and keeping their eye on the ball, as my dad's sagely advice instructed when I was playing baseball, and he's absolutely right, of course. But if batters confronting pitches at these speeds were relying solely on their eyes, it'd be physically impossible for them to actually ever react rapidly enough to swing before the ball was already in the catcher's glove. And so how is that possible, right? Well, it's really a topic for another day. It's an interesting topic, but the point is that we clearly have a whole broad repertoire of systems that enable us to both detect and physically respond to events in our environment that don't require conscious awareness. And so one of our unconscious processes, or many of our unconscious processes, are capable of some pretty impressive feats. And it's a beautiful example, in my opinion, my humble opinion, and that's something that Eagleman is gifted at creating. Also, in case you're wondering, the fastest pitch of all time reached just over 105 miles per hour, clocked by Guinness Book of World Records, with which I know you are familiar, Daryl. And so that's about 169 kilometers per hour. Pretty bananas. So, by the way, I can't help myself. I actually met David Eagleman several times when I was at Baylor College of Medicine in Houston while he was a professor there. He's a really nice guy. I'd actually just started participating in an effort that he'd started, exploring a kind of like still emerging field called neuro-law, which basically confronts the increasing role that science, and particularly the science of consciousness and the brain, neuroscience, is playing in the legal system, and particularly criminal law. And I'd gone to 2 meetings that he had at his lab, and I was like getting pretty psyched about the whole project, trying to like figure out how I could plug into the effort as basically the only like cell molecular circuits person in the room, full of cognitive neuroscience people, computer science people, and lawyers. But then I ended up, heading up here to Philly, where I ultimately did my PhD. And as evidence, this copy of Incognito happens to be signed. I don't know if you can see that. Yeah, you can sort of see it. Anyways, I definitely recommend this book for anybody who's, just like a fun tour of some of the fun parts of neuroscience. Thank you very much, Claudio, for the award. And hey, Georgia. So everybody, if anybody's unfamiliar, Georgia Auteri is a rising PhD. She studies evolutionary biology, focusing on bats, but has a much broader base of knowledge. And she's active here on this platform. So if you're interested, check her out, Georgia Auteri. Okay. So that whole jaunt down memory lane was at least intended to illustrate 2 things: how incredibly complex and amazing our visual system is, as well as how incredibly integrated and adaptive our broader sensory systems and consciousness are, enabling us to respond to changes in our environment using vision, along with a whole repertoire of other capabilities to respond to those changes even faster than our visual system can allow for, right? According to the laws of physics. So a question that I've always had, that I had before going into neuroscience has always been, what is it like to be blind? Do folks without typical vision just experience complete darkness? And the reason that I wondered that was based on what I would experience in a very dark room with my eyes closed, even deprived of, any source of visual stimulation. I could still sort of see things. I could, and I still can, see little flickers of what seems like light. And it isn't like someone pointing a flashlight at, my eyeballs with my eyes closed, right? The light has a whole variety of colors, red, green, and blue, generally speaking, combinations of those. And it occurs, those patterns occur, in what seem to be like geometric patterns, right? Sort of random patterns, but patterns nonetheless. And, it's an interesting experience. And I bet you've experienced that as well. So that visual experience, in the absence of any actual photons splashing up against your eyes, is called phosphenes. And it's a perception of light in the absence of any actual light. And it's absolutely normal and natural. And it's essentially the byproduct of neurons in the part of the brain that process vision becoming spontaneously active, starting to sort of randomly fire impulses, getting sort of kind of randomly. And so, this, of course, presumes that the visual cortex as well, and we'll talk about, where the visual cortex is in just a sec, but this presumes that the neurons in the visual cortex are still intact. Which speaks to what you were bringing up, Daryl, that there are different kinds of blindness, right? But the point is that, even for folks who qualify as completely blind, right, no light perception whatsoever, those neurons can still be there, but they're just not receiving that sort of eyeball-based stimulation. Hey, Helen, good to see you. Right, do blind people experience visible blackness? Right, well, we'll get into that. We have a fun story from a journalist, actually, who's blind. And so it turns out that there are definitely many causes of blindness. And to be clear, but it turns out that at least some people who are blind do not experience pure darkness. Rather, there's the perception of, and I'm quoting from somebody who has this experience, there's a perception of, quote, bright, colorful, ever-changing, often terribly distracting light, end quote. So that's a quote from an article written by somebody who's designated as having no light perception, right? Totally blind. And I've linked, I have a link to his article if anybody's interested in checking it out. Happy to post it. I totally recommend reading it if you've ever wondered what I did. And so it's interesting beyond just his description of purely what visual perception is like for him. So definitely recommend checking it out. So, okay. So that's what makes this achievement that this group has demonstrated so impressive. And so this is work coming out of a part of the world called Elche, Spain, in Spain. Which, by the way, I don't know about you, seems like one of those outrageously beautiful parts of the world where people get to do like actual science in paradise, right? Like there are some solid neuroscience labs in Nice, France, for example. Or I imagine UC San Diego is another place that's just, I don't know, I can't imagine getting any work done, when outdoors is just like perpetually perfect and beautiful. But anyway, so that's where this group is, at Miguel Hernandez University, specifically in Paradise. And so there's a wonderful article in MIT Tech Review, to which I'm happy to link if anybody's interested, which explores this story in a sort of informal narrative way. But before we get too into it, I think it's worth noting that at the outset that I think there have been a lot of like exaggerated headlines about this innovation, like implying that they've cured blindness, and that's absolutely not what this is. But it is still really interesting. And so fundamentally, what this group has done is to show that we can implant a brain-computer interface, right, which basically has like a bunch of tiny little electrodes, little things that can stimulate neurons, making them fire, record from neurons, listening in on them. They can implant those electrodes right in the part of the brain that enables us to understand what our eyes are seeing. And most interestingly, they did it with a human participant. And that's who this very generous human is, Bernadetta Gomez. So that's her wearing her, the whole sort of preliminary sort of beta level device, right? Which basically just looks like some sort of futuristic glasses. And they obviously have a bit of extra equipment going on just beyond like just some special lenses or whatever. But here is what that device really looks like. Here, maybe we'll do that. Okay. And so the glasses have a camera right in the middle, right in the middle of the bridge of the glasses. And then that sort of fancy camera sends information to a device that then processes the images through some filters and stuff, processing the video essentially. And all of that ultimately ends up at their electrode array, which is this is like the actual meat and potatoes of the device. And so they call this Serostim 96. And so astute listeners probably noticed that 96 number there. And so believe it or not, that's not just some like random large number that they're including to make their device sound particularly advanced. Like this is better than the 95 version. It's not like the Vision Generator 5000 or whatever. The 96 refers to the number of electrodes on their electrode array. And so interestingly, so they theoretically had all of those electrodes at their disposal, 96 of them, but they could only use 16 of them at a time to stimulate neurons in a part of the brain that processes visual perception. Okay, so let's talk about where that is. Yeah, here we go. Okay, so that is the visual cortex, right? And so we have all of these photons splashing up against our eyeballs and all of this neurophysiology, loopy neurophysiology as Eagleman calls it. It all has to not only process all of those patterns of photons, patterns that are literally changing at the speed of light, But then all of that information has to be transmitted to exactly the opposite side of our brain. Sort of a silly design if you think about it. But anyway, so that's their device. And so, yeah, share the article. Okay, here, let me do that right now before I get too far afield. Boop, boop, boop. Here we go. I'll share just the link rather than the citation because I feel like it's easier for you. And you can check out the citation information if you're really interested. But yeah, let's, so, and then, oh, Claudia says, my post-stroke lateral blindness feels like a nothing zone. Oh, yes, hard to define it. Sometimes I think it's a dark area as when I'm waking up, but most often my focus is on the active vision. And the left side blindness is neither dark nor bright, just blank as the end of a normal visual field. Does that make sense? Yes. So for anybody who is unaware, Claudio unfortunately recently had a stroke, and he lost some of his vision. And so what's interesting about that, Claudio, is like, so the way the eyes are organized, it's actually a little bit more complicated than I think a lot of us study in maybe, I don't know, intro biology. It's not like, you know, as is the case with a lot of our bodies, you know, this eye is regulated, controlled by the opposite side of the brain. Sort of like, you know, the way that, you know, this side of our body, the muscles on this side of our body are controlled by brain regions that are on this side of our brain, opposing sides. But actually, it's a little bit more complicated than that when it comes to eyes. It's actually that this half of this eye is controlled over here. You sort of you split the eyes in half, and the entire eye doesn't all go to the same opposing side of the brain. It's kind of ridiculous, right? It's kind of crazy. But so as a result, depending on where specifically, you know, a stroke can occur, for anybody who doesn't know, a stroke is essentially, there's a couple different ways to have a stroke, but fundamentally what it is a deprivation of oxygen to a part of the brain. And that can happen essentially anywhere. And depending upon where that happens in the brain, you can get different manifestations, right? You can get a loss of motor activity, paralysis, essentially. Or you can get loss of language capabilities where somebody finds it very difficult to speak. Maybe they'll slur their words, or they just won't be able to speak anymore. You can get a whole bunch of things. You can get changes in mood and stuff like that, or you can get loss of vision. And generally speaking, because, you know, the brain, it's not like it's either or. You either lose it or you have it. Oftentimes it's sort of like, it's just a sort of diminished capability, you know, regardless of what the capability is. So you can still have some vision, you can still have some motor activity. It's just not as robust as it was pre-stroke. And so, yeah, what you're describing, it is interesting. I mean, of course, it's hard to really understand without actually experiencing, like a lot of human consciousness is. But like, you know, one way that folks who are fortunate to not have lost vision to sort of probe what that's like is you can actually detect where there's a part of our visual field that's actually a dead zone. And that part is where the optic nerve basically connects to our retina, right? And so there's like, you know, we have these photoreceptors. I didn't expect to go into eye biology so much, but I wish I'd come a little bit more prepared. But the way that we can see things is that we have these photoreceptors that are just proteins that are stimulated by light, different wavelengths of light. We have some photoreceptors, proteins that are stimulated by essentially any light. Those are called rods. And then we have photoreceptors that are stimulated by certain wavelengths of light, specifically red wavelengths, green wavelengths, and blue wavelengths, right? And so, you know, photons can travel at all different, you know, types of wavelengths, and depending upon which wavelength, that'll correspond to a certain color. And so we, you know, we have these photoreceptors, right, in our eyes. They're actually pointed towards us, which is kind of ridiculous. I've talked about that in the past, you know, as opposed to like pointing outward where the light is coming from, they're pointed inward at our brain, which is like, so the light actually has to like come into our eye and then bounce off the back of our retina and then hit the photoreceptors. Yeah, it's just kind of, there's actually some interesting arguments as to why that might actually be superior to that, like that organization might be better than just having them pointing the way, if you were to sit down and design an eye that looks like ours, you'd probably, you know, turn them around so they're pointing towards the source of the light. But anyway, so we have these photoreceptors, right? And they cover most of our retina, and, you know, certain types of photoreceptors are more densely packed in certain regions of our eyes, of our retina. But there is a certain zone in both eyes in which there are no photoreceptors. It's a dead zone. That's where the optic nerve connects. And you can actually, and normally you never notice that. You probably didn't, if you've never heard of this, you probably never would have even thought that that was a thing, because if you have full vision, then you never notice it, but you can. So maybe I can use my daughter's artwork. I don't know if you probably can't see that because it's too bright. Oh, you can see it if I go closer. Yeah, pretty nice, right? She calls this a banana. That's her version of a banana. Very impressive, if you ask me. But anyway, so if you, the classic way to do this is you have like a circle and a plus sign right next to each other. And usually it's like a very simple, it's like black and white, circle, plus sign, white, or, black, plus sign. And what you do is you cover one eye and then you move closer or further away to like to the plus sign or the circle sign. You're supposed to focus the open eye on either the plus or the circle sign. And, once you reach a certain distance, what you ultimately can see is that symbol disappears. It's not even there. But interestingly, what your eyes do is or what your brain does is it fills in that gap, right? And so it's not that there's just like a circle of darkness, right? Which is sort of what you might think because there's no light coming from that area. What the brain does is it sort of interpolates, it sort of extrapolates, and it basically like takes the visual information from around that dead zone and just superimposes it over the dead zone. And so what you see, let's say you have a black plus sign on a white background, and you get to that dead zone, that visual dead zone, then all you see is just white, right? As opposed to a little dark circle. So what you're describing, the reason I went into that whole laborious, tedious explanation, Claudio, is like, despite the fact that it is a dead zone, there is no visual information there, we don't experience it that way, right? We experience it more, it's not there, and we wouldn't have noticed that it was there, right? It doesn't feel like we've lost something, right? It doesn't feel like we've lost something. It feels just more like, oh, this is just normal. And, I'm sure that departs quite a bit from your experience, obviously, but yeah, I can understand that it wouldn't be so simple as just like something was there and now it's gone and I miss it. It's slightly different, right? Anyways, yeah, we'll get into it a little bit more. And then Helen says, my mom has lots of complications due to diabetes, including retinopathy. And she tells me all of this weird patterns like spider web across her field of vision, or floaters and weird light patterns. Yeah, exactly. That's what we're talking about. That's sort of similar to phosphenes, right? Where if you close your eyes and, this is also true if you like rub your eyes a little bit, not super hard, obviously, but if you rub them a little bit, you can sort of like crank up the volume on those phosphenes. Certain substances really amplify phosphenes. And so when, somebody under the influence of a psychedelic, hallucinogen, or cannabis or something like that, when they close their eyes, the magnitude or the brightness, I guess, of those phosphenes is sort of turned up. Interestingly, some over-the-counter substances also do that. Just like a camera sensor. Oh, is that right? I didn't know that. Tim, good to see you, man. Hope you're doing well. And I appreciate the award. Thanks, man. It's been a while. Are you still in New York City? I'm not sure. But okay, so, and then Claudia says, I detected this natural blind spot in my right eye by chance and rushed to the hospital believing it could be another smaller stroke. Oh, interesting. Well, I'm glad that it wasn't. And I'm glad that you're paying attention to that, because strokes can happen in that kind of succession. Yeah. Okay. So all right. So we have our visual cortex there, and so yeah, and so all of this visual information coming from our retinae have to travel all completely, literally across the brain to then finally land at our visual cortex. And so that. And that's where their device is implanted, in the visual cortex, in the occipital lobe, right at the back of the brain. And so they were working with Bernadetta, right? And so she had been blind for 16 years. She's 57 years old, and she lost her vision at 42 due to damage to the neurophysiology that connects the eyes to the visual cortex. And so, while she was born with vision, she lost it. And it was lost to the extent that she was completely incapable of detecting any light. Thank you very much for the award, Tim. I really appreciate it, man. Okay, so Bernadetta has this electrode array temporarily implanted in her occipital lobe, right at the back of the brain, and she was capable of seeing light for a 6 month period. And so I'm going to quote from that wonderful article in MIT Tech Review. Quote, using this implant, Gomez identified ceiling lights, letters, basic shapes printed on paper, and people. She even played a simple Pac-Man-like computer game piped directly into her brain. 4 days a week for the duration of the experiment, Gomez was led to the lab by her sighted husband and hooked into the system. How cool is that? And so there's another quote from the article that illustrates how counterintuitively like casual the implantation of the whole process of implantation is. Oh wait, yeah, okay. And so Gomez is sitting in a room, right, with the scientist who devised the device, and there's another person who's blind in the room, evaluating whether or not they'd like to participate the way that Bernadetta did. Bernadetta did. And so, quote, at one point during this meeting, as Fernandez, the lead scientist, explains how the hardware connects to the skull, Gomez interrupts the discussion, tilts forward, and places the prospect's hand on the back of her head, where the metal outlet used to be. Today, there's virtually no evidence of the port. The implant surgery was so uneventful, she says, that she came to the lab the very next day to get plugged in and start the experiments. She had no problems or pain since. That's pretty awesome. Like, I would absolutely expect that there would be at least a bit of a recovery process, like ice cream and comfort movies for at least a week. I would demand that. But anyway, so this group based the way that they stimulate using that implant on an interesting strategy. So they were learning how the human eye sends signals to the visual cortex by literally measuring signals from the eyes of recently deceased organ donors. Because it turns out that the human retina can only be kept for about 7 hours. They had a lot of like late night calls to rush to the hospital or whatever. And so because it can only really stay viable for about 7 hours. But anyway, so that's how they figured out how to stimulate the visual cortex to achieve their desired stimulation, right? And so Bernadetta was able to decipher letters and basic shapes with this implant. And so you can see no, not that one. In this one. That's how my daughter would say it. This one. So you can see here. I'll make it a little bit bigger so you can actually really see it. And so on top of this figure, you're literally seeing what which of the electrodes we're sending activation, right? We're stimulating the neurons close to them, near the electrode in Bernadetta's visual cortex. And so you're seeing the difference between a capital O and a lowercase o. And as you can see, over time, Bernadetta grew to get better and better at discriminating that subtle difference. I mean, obviously, these are different characters, but a capital O and a lowercase o, that's pretty subtle. We're talking about like millimeters of a difference in what those characters look like. So, and she reached 100% accuracy after just a week. That's pretty awesome. And so they did a kind of like real-time test and this is pretty fun. To see how effectively Bernadetta was able to discriminate between stimulation on the right side of the implant and on the left side, right? And so it's pretty fun. Here we go. Okay. And so fundamentally what you're going to be seeing is Bernadetta sitting down with her eyes closed while she's plugged into the system. You're seeing a black box on top, right? And then Maggie Simpson from the TV show The Simpsons periodically appears and fires a shot at the camera, let's say, or at you, right? And she's either using her right hand or her left hand to do it. And so depending on which hand she's using, the implanted electrodes will stimulate certain neurons that correlate with right or left. And so then, as soon as she can, Bernadetta presses a button for, either right right or left to get a sense of whether the implant was able to successfully and consistently experience or deliver the experience of that visual perception of, being on the right or the left-hand side. And so, if I'm reading the study correctly, it's not like she was actually literally seeing Maggie Simpson, right? That's really just for our benefit, I'm pretty sure. It would be more like just a sort of like flash of light, a transient, short little flash of light, right? And then, but Bernadetta could learn, the flash of light is here or it's here, right? And so that's basically enough to perceive things like letters, sidewalks, hallways, just very general visual experiences. And that can for sure be super helpful. But, keep in mind, this was just one teeny tiny little implant. Here, I should have brought this to your attention when do you see that? So that's the implant. Again, that's the micro electrode array. You see at the bottom of that image, you can see like this black line. That line is to indicate what 2 millimeters looks like, right? So this is like very zoomed in on the implant. It would be really minuscule. 2 millimeters is quite small. And she had just one of them. So imagine implanting significantly more of them. That's a sort of Wild West, right? Because it's hard to know at what point the artificial stimulation departs so significantly from our natural visual system that it starts to seem just sort of like hyped up phosphenes or like, TV snow. It's sort of like when if you crank up the volume on your laptop speakers or speakers that can't really handle high volume, and it just starts to go like, right? It just gets so distorted that you can't even understand what's being played anymore. Maybe it's sort of like that, where if you implant so many of these things, let's say, Bernadetta had just one, imagine 5, imagine 50, imagine 500, 5,000, right? There could be a point at which you just start to get, and your visual system, your visual perception is just sort of like that TV snow, which doesn't ever really happen anymore because nobody uses that kind of a TV. But anyways, if you're of a certain age, you might remember that. And so Fernandez, okay, and then, sorry, so it's tiny. Yeah, it's tiny. And so Daryl says, I imagine even the most trivial visual signals would be hugely beneficial to somebody who's completely blind. Absolutely. Yeah, exactly. And so the group describes this as being sort of like being able to see a 10 by 10 pixel by 10 pixel kind of visual experience, which is, pretty minuscule. Depending on where this video is landing right now, this is about a 1,280 by 720, right? And so 10 by 10 is like pretty minuscule. But, again, that's just one of these teeny tiny little implants. And so imagine 5,000 of them. All of a sudden, you're starting to get some real resolution. You could perhaps, discriminate even more complex and nuanced visual fields. And so Fernandez, the lead scientist on this project, makes what I think is a perfect analogy. So this device is to vision as a cochlear implant is to hearing. Cochlear implants are essentially just microphones, like the one that I'm using right now, that are wired directly into the inner ear, bypassing, the part of the auditory nervous system that normally enables us to hear things and just directly stimulating the part of the brain that detects sound. That's a cochlear implant. They've been around for a very long time. And they work very well. It enables people who would be otherwise completely deaf to have conversations, right? And so that's essentially what's going on here with this implant. Evidently, Bernadetta would have liked to have kept the implant longer than the experiment permitted. And she's also, which is pretty adorable, she's planning on framing and hanging her implant right on a wall once the team of scientists is done studying it. I'm sure they're studying it to get a sense of like how much the materials of the implant degraded over that 6-month period, as well as, perhaps any kind of like footprints or fingerprints from Bernadetta's own immune system that, might have been left on the implant. Basically, anytime there's any kind of foreign body inside of our body, our immune system will respond. We just, broadly speaking, call that inflammation, but that's the immune system reacting to this thing that should not be there, or doesn't have the sort of like, the hall passes that are necessary for something in the body to not be attacked by the immune system. It's a complicated story, but basically, so that's inevitable. That's anticipated, and you can take various steps to mitigate that. Various coatings, for example, can kind of make it just more difficult for the immune system to even recognize or interact with it. But and so I'm sure that's part of what they're working on before they give it to Bernadetta for her, what I think would be a very, very cool bit of art. But anyways, given this group's success, it turns out that they already have plans to work with 5 more people who are blind. Pretty exciting. And so remember that article that was written by someone who has a blindness that's designated as no light perception, right? Totally blind. Well, part of the reason that his article is so insightful is that he gets into the reasons that his blindness is debilitating beyond purely the inability to see, right? Which is in and of itself is pretty debilitating. And so, as I noted, he describes the constant perception of spontaneous geometric patterns of light despite being blind. The constant perception of it, right? Think about that. It's constant, meaning it's inescapable. So he can't close his eyes to blot out the perception. It's just always there. Can you imagine something like that? Like, try to imagine never being able to stop seeing something. Hell, like try to imagine any sensory experience that you just could not stop. Imagine an odor that you could not stop smelling, or even just a source of pressure somewhere pressing up against your body that you could not stop feeling. Or imagine a sound that you couldn't stop hearing, like tinnitus, for example, which is can be quite terrible and debilitating for some folks. So I don't know about you, but before I'd really explored accounts from people like Damon Rose, who wrote that article about what it's like to be blind, at least for him, I hadn't really contemplated those sort of secondary and tertiary ramifications of losing vision. I hadn't really thought about the challenges that they face beyond just having to adapt to not being able to see their surroundings. And so Georgia says, after you implant it, is it very difficult to undo? Wait, after you implant it, is it very difficult to undo? No, that was part of the experiment. She only had it in for 6 months. So, I mean, keep in mind, these are really, really small. And, I should also note, like, maybe this is obvious for folks, but maybe it's not. Like, again, that black bar is 2 millimeters. So this is a very, very small implant. And as a result, those spikes, those electrodes are also very, very small. They like just barely penetrate into the brain, the outermost layers of the brain. And so you might be concerned about damage. Like pretty much anytime you're moving something in the brain, neurons are being damaged, right? It's inevitable. It's something that I struggled with during my PhD. Very frustrating, and not enough scientists, in my opinion, accommodate that or interpret the results with that in mind. But anyways, so the damage from these is very, very, very minimal, right? You're probably breaking some capillaries. You might be damaging a couple of neurons here or there, but hey, who's counting? No, but like that's not a very significant insult to the brain. So all this to say, it's not like implanting an electrode deep into the brain. That is done, right? Deep brain stimulation has been used to treat a whole variety of things, most notably Parkinson's disease. But those electrodes are really going down deep into the brain. These are just like hanging out right at the outer layers. So not a very big deal. So yeah, all this to say, it's not very hard to undo. Yes, it would drive you nuts. Exactly, Helen. Yeah. So I personally, and maybe it's just me, I hadn't really contemplated like what it would be like to not be able to just close my eyes to just escape a visual experience of some sort. Like that's just sort of mind-boggling. But that's why I found this research so compelling. It's not only helping individuals who are living without access to a part of the universe that like quite literally delivers the color of our surroundings, light, but it's also some of the early steps towards helping to anchor their sensorium, right? Such that their visual perception isn't just the byproduct of the essentially random whims of the neurons that live back here, generating those random geometric patterns of light. Now, with something like this, their visual perception can be anchored. And theoretically, they might be able to control or exert a little bit more control over their visual experience. All right, that's what I have for you today. If you dig this kind of discussion, neuroscience and biotechnology and stuff like that, maybe you'll consider subscribing and liking and pressing all those helpful internet buttons. Otherwise, have a great week, and I'll be here next week with another interesting instance of progress in biomedical science. Okay, and so Daryl says, so neurons will randomly fire without sensory stimulus. Yes, that's correct. That is so Daryl, you might be familiar with like sensory deprivation tanks, right? So for anybody who's not familiar, those are basically like small, not super small, big enough to house a human pretty comfortably, chambers with water that's so salty that you'll float, right? That'll make you buoyant. And the water temperature tries to achieve a temperature where it's difficult for you to distinguish between like the air and the water and your body, right? So you start to sort of lose that anchoring of like, I'm in a room that's too cold or I'm in a room that's too hot, right? And it's also entirely dark and entirely silent, right? So the idea is it's sensory deprivation. You're depriving your central nervous system of sensory inputs, and those sensory inputs anchor us to reality. There's some interesting conditions where that anchoring capability of our sensory systems is lost, and that can be associated with schizophrenia, for example, or psychosis. But anyways, the visual system deprived of any incoming external sensory stimulation, that's what happens. No, I'm sorry. In the sensory deprivation tank, people, and I've experienced this, people describe these very exotic and strange, almost hallucinogenic experiences where it's clear that their brain is just like running wild. Right. You can have all kinds of wacky memories. You can have visual experiences. People do quite literally hallucinate. And that's part of why, because you're in a situation where your brain, your consciousness, your experience is no longer anchored by that external source of information about what's going on around you. And so the brain, which always does, part of like how the brain functions is to predict events in its environment. Right. That's essentially how we navigate the universe. The universe, I guess, but particularly our lives, is that, your brain develops expectations, makes predictions about what's going to happen in your environment. And then when that prediction departs from the actual outcome, you have an emotional experience, which is either positive or negative, right? If it's positive, it's something happened that you didn't expect that is good for you, right? When it's and that feels good. There's a bit of dopamine literally associated with that. And then when something happens that you didn't expect that's bad for you, then the opposite happens. It's actually a complete plummeting of dopamine levels. And we experience that in the form of emotions, right? It's also how we learn things, right? That's why testing and quizzing and that kind of stuff is so important to actually learning something. And our sensory systems work the same way, right? Where, we can't literally take in all of the information about our visual surroundings and know what's happening, despite the fact that we see a lot of stuff, right, in our field of vision. We really only focus on a small component of our visual field, right? It's specifically where a part of the eye that's called the fovea is pointing. And that's where our conscious awareness is really focused. We can see things happening over here, but they're kind of in low resolution, right? And you can sort of like tickle the contours of the sort of the extent, the boundaries of your visual field. Sorry, I was like staring at the camera while I was doing that for myself. But literally, like the photoreceptors, the biology that is devoted to that part of our visual field are less sophisticated, are less precise. And so in any case, yeah. When our sensory nervous system is making predictions about all that other stuff that's happening around us, and then when we're surprised, then we detect it. Then our conscious awareness is focused on it, right? And so when you're in a situation where you're like in a sensory deprivation take, for example, those predictions are just, there is no comparison, right? And so they're just left to run wild. And the same kind of functional event is going on here, where somebody who doesn't have access to that external anchor, their visual cortex just will run wild, and they'll have all kinds of interesting geometric experiences, right? And then so Cosmic Rose says, I know a blind streamer that cooks meals and sews during a live broadcast. Give her this stream to play. Is there an article she should access for info on the topic? Yes, here. Here we go. Let's see. The best one is probably this one. Hello? Are we not cooperating? Here we go. Yeah. The MIT Tech Review article is probably the most like approachable. I mean, I'm happy. Oh, I guess I did link to the actual study, but the study it's not as fun because they're just giving you the science. This MIT Tech Review article interprets it a bit for you. MIT Tech Review is really good for that. Yeah, it would be really interesting to hear what she says. Oh, and then another one that would be interesting maybe for her is this one. This is the article by the journalist who is completely blind, describing what his experience is like. And I think it's in BBC. And then thank you very much for the award, Hugh. I really appreciate it, man. She uses TTS for comments, and we have fantastic convos in different streams also. Interesting. Yeah, that's really cool. Sewing, that's pretty impressive. I can't do that, and I am sighted. Link not showing on YouTube. Oh, I always forget that. I'm sorry, I'm such a dummy. Here we go. Yeah, I put that in the wrong. Technology. Here we go. So we'll do boop. Can I just? Well, okay. Here is here's the BBC account, the account of somebody who is blind, describing it, and then here's the MIT Tech Review article. This is really the one that I think she should read. I mean, she can read all of them if she wants, but here. There we go. I've seen a blind YouTuber, Daryl says, who will argue that blindness is not even a disability, but of course he was 100% blind from birth. Nice guy, though. He has no reference. Yeah, exactly. Yeah, I mean, like it depends on it. It's semantic at a certain point, right? But yeah, it is a really interesting dynamic where I've also heard similar things from people who have synesthesia, which seems very distracting at a minimum, if not debilitating. They will describe it. I mean, in some cases it can be, right? But they'll describe it as like, this is just what I know. Like it's not like I had something that I lost, or I didn't have this synesthesia and now I have it. And all of a sudden I have to like completely restructure my life around this. And so yeah, I mean, that doesn't surprise me. I mean, but I think objectively, right, we have devised a world in which the assumption is that people are largely, neurotypical, let's call it, where you have vision, you have your ability to hear, you have the ability to walk, you have the ability to do all these other things. And so as a result, I mean, it's good, of course, that folks can adapt to that world. But, as somebody who's like engaged in policy at this point, like I think we could do a much better job in making the world more hospitable to people who don't have all of those capabilities, or their capabilities are just different. And so, power to them. If they don't see it as a disability, I bet that they're better at a whole variety of things than I am, right? But regardless of what you want to call it, I do think that we still have some progress to make regarding making the world more accessible for people who just have different nervous systems. Yeah, Cosmic Pros, I would appreciate that. Please do. All right, I guess I'll call it there. Thanks for joining, everybody. Once again, if you dig conversations about biology and neuroscience and consciousness and stuff like that, maybe you'll consider subscribing and hitting all those happy internet buttons that help tell the algorithm that I'm not evil. And otherwise, have a great week, and I'll be back next week. See you, everybody.